today we're going to be talking about HIV, or the human immunodeficiency virus. This virus is called human immunodeficiency virus because it makes our immune system deficient. How does it do it? HIV attacks human immune cells, or CD4 T cells, and macrophages, and then causes a chronic blood-borne infection. HIV is an important virus to study because there are 87 million people infected or dead worldwide ever since the pandemic started. Additionally, in Australia, there's over 25,000 diagnosed with HIV. These include sex workers, indigenous populations, drug users, and heterosexuals. There are also 10,000 undiagnosed. So clearly this is a virus which has an enormous global and national, which places an enormous global burden as well as a big national burden. And it's something that we should take the time to consider and learn about. Now, HIV belongs to the family of lentiviruses. Lentivirus, because lenti means slow, and again it's a virus. These viruses belong to the retroviruses family because they're single-stranded RNA molecules, positively stranded, which cause a delay in the onset of symptoms. Delay, slow. There are two types of HIV, HIV1 and HIV2. HIV1 descended from the HIV1 is also known as the simian immunodeficiency virus, chimpanzee, because it was first found in chimpanzees. This HIV1 infects 95% of cases worldwide. Now in HIV2, this makes up less than 5% of all infections. They were derived from soothing manga bee monkeys. Hence, another name for them is SMM. And the transmission from animal to humans is believed to be from the consumption of bushmeat. Okay, now that we understand the family, let's look at the risk factors for contracting HIV. So, the risk factors include large sexual networks. So this means sex workers are at higher risk. And it also means overlapping sexual partners, age mixing, all these behavioral risk factors. Around 80% of HIV cases are actually acquired from sexual activity. Other risk behaviors include little or non-condom use, anal sex because the thin rectum lining allows HIV to enter more readily, low rates of circumcision, high STD rates, high viral load, and drug injections are other risk factors. Okay, now we're going to look at the structure of the virus. So, the HIV virus has many different coats, and different proteins make up each coat. However, there are three main segments within the splice RNA of the HIV virus, which are called GAG, POL, and OMV. Now, the OMV RNA segment encodes for proteins GP120 and GP41, which are shown here. One is on the cell surface to allow for binding to CD4 or macrophages for infection. The other one is just the transmembrane fusion domain. Encoded by the GAG RNA segment are the proteins P7, P24, and P17. They play roles in the integrity of the nucleocapsid, capsid, and matrix respectively. Encoded by the po-RNA segment are the proteins P66/51, P32, P11. These are all enzymes which play a role in ensuring correct RNA replication takes place, or proteins are translated. And as you can see, they're all labeled here once again, so you can have a look. GAG encodes for structural proteins in the core, matrix, capsid, and nucleocapsid. PO encodes for enzymes such as proteases, reverse transcriptase, RNase H, integrase, etc. Proteins encodes for the envelope glycoproteins from spliced mRNA. Now, it is important to note that a high degree of variability exists for proteins, which are GP120 and GP41, and a high degree of variability also exists for GAG proteins, which make up the nucleocapsid, capsid, matrix, and core, etc. This is important to note because it will make us understand what makes HIV such a deadly pathogen. Now, we're going to be talking about the replication cycle of HIV. HIV actually goes through 8 stages of replication. The first stage is attachment, as shown here. So we can see the GP120 protein attaching to a CD4 receptor with the assistance of chemokine co-receptors CCR5 and CXR4. If we look at this process in more detail, we see that the GP120 exists as a trimer, after which, upon CD4 binding or binding to the CD4 receptor, there are structural changes to the GP120 which allows for access to the binding site. Subsequently, the co-receptor could then bind the co-receptors, which are chemokine receptors, either CCR5 or CXCR4, increase the GP41 fusion and consequent insertion into the CD4 cell or macrophage cell membrane. The GP41, as previously mentioned, is just this area here that plays a role in fusion. When the membrane does fuse, the shape of GP41 changes, and this allows for fusion. And they form somewhat of a 6-helix bundle. Now, in the early stages of HIV infection, you have greater CCR5 binding, but in later stages you have greater CCRX4 chemokine receptor co-receptor binding. Now, after the attachment and fusion, which we've already both discussed, it undergoes reverse transcription. What happens in the reverse transcription phase? Well, reverse transcriptase converts viral genomic RNA into proviral cDNA. This is a process that has a high error rate, 1 to 10 to the power of 4 mistakes, 1 to the 10 power of 4 nucleotides copied. So the cDNA is different from the RNA because it is single-stranded and it is all complementary, and without the cDNA, the virus cannot insert its genome into the host. So reverse transcriptase, it also duplicates the sequences at the ends of the viral RNA. So you can see its action over here. So generally it would look like what is written down in black, but upon duplication you see that the 3-terminal repeat is now at the 5', is copied to the 5', and the 5' U5 is copied to the 3' end. So with reverse transcriptase, you now have a proviral cDNA complementary DNA segment, which can then enter the nucleus and become integrated into the host genome or the host cell DNA to be transcribed and made into different proteins for assembly. However, before we move on, let's just talk about the integrase enzyme. So this can occur in both resting and terminally differentiated cells. It catalyzes the integration, the random integration of viral cDNA into cell DNA. Yep, so after that you have transcription. For transcription to occur, it is important to understand the 5' long terminal repeats. So these are created when the reverse transcriptase duplicates the sequences at the ends of viral RNAs. The 5' LTR has a promoter region, which one of its proteins called TAT binds to increase expression. It also silences its expression after initial replication so as not to be detected readily by the immune system. And it responds to cellular proteins made during the T cell immune activation. Now, what do we mean by this? Okay, so this again is a drawn diagram of the integrated provirus. So this is the 5' long terminal repeat that we see here. It has enhancer sequences, transcriptional start signals, and tap sites. It allows for it responds to cellular proteins made during T cell immune activation. In the activation, because the U3 segment has an enhancer component, core enhancer in common, which allows NF-kappa B to bind and activate it. It also has other modulatory elements and promoter regions for this to occur. And over here we have the 3' end. Okay, so now that moving on to transcription, it makes all these splice mRNAs, and then there's also the translation of proteins, which are vital, and these are then assembled as shown here, which then bud out of the cell and become mature HIV molecules ready to infect again. However, before we move on, let's try and understand the extra proteins that HIV encodes for to make it so deadly. So we have here a diagram excluding the GAG, POL, and OM descriptions. First of all, let's talk about TAT. So TAT binds to the TAR region, which is shown here. And this starts the HIV transcription. Then there is REV, which is shown here, which regulates gene expression through binding to the RE location, which is shown here in the OMV RNA segment. And this essentially helps to transport HIV RNA to the cytoplasm, which may be unspliced. And you need these to be translated to the cytoplasm because you need translation to occur. Okay, so now that we understand the replication cycle, we are going to discuss how HIV becomes this pathogenic disease that it is. So upon mucosal exposure to HIV, there is selective infection by R5 strains, which means that you would have, although all HIV molecules would bind to ssRNA on macrophages, some will bind more favorably, and because of the great amount of variation that exists, the best surviving HIV or the strongest binding HIV will persist. Then the HIV binds to the dendritic cell, while via the CD209 C-type lectin receptor. And using this binding, it travels with the dendritic cell to the regional lymph nodes where they spread to CD4 plus T lymphocytes, and again you infect them via GP120 binding to CCXR4. Then they enter into more cells in the bloodstream and disseminate. Okay, now we're going to talk about the progress of untreated HIV. So essentially you have 3 stages: primary infection, clinical latency, and symptomatic infection. Primary infection actually only lasts a year, and in here you have plasma viremia peaking at around 6 weeks before dropping again. This period may be accompanied by a peak in maximum decline in CD4 plus T cells and CD4 plus T cells in the body and the blood. And this period may also be accompanied by acute HIV syndrome, wide dissemination of the virus, and seeding of the virus into lymphoid organs. So in the primary infection stage, you may also experience symptoms such as muscle pain or myalgia, joint pain or arthralgia. You can experience oral thrush, nausea, vomiting. Your lymph nodes can swell up. You can have fever, headache, pharyngitis, headache, etc. There's also more of a systemic effect, as in weight loss or lethargy. And during this primary infection period, the virus incubates for about 2 to 4 weeks, and then develops disease for 1 to 4 weeks. Unfortunately, during this stage, it kills all CD4+. It kills a large majority of CD4+ and CCR5+ memory T cells, which may be activated or resting. And it also leads to lots of CD4 substrate exhaustion and breaks epithelial channels, but we'll discuss this in more detail later. Yeah. Now you have, subsequent to that, you have clinical latency, which is again asymptomatic. Then you have symptomatic infection, which may constitute symptoms and lead to opportunistic infection. But now we're going to discuss even more detail what happens in that primary infection period. So over here we can see a diagram of HIV antigen and HIV-1 antibody over 60 days post-infection. and what tests you can perform to confirm the presence of an HIV infection. And we can see that the earliest test we can conduct is a viral RNA PCR test, but this is taken after 20 days after the initial infection. The next best test is the P24 antigen test, and this is taken around 28 days. The P24 is a capsid protein, as previously mentioned, in the virus, so that's a possible option. Then you have testing for antibodies, but antibodies don't develop until 35 days after the primary infection, and then it dips a little bit, the HIV-1 antibody, so there's a period where this test enzymatic immunoassay isn't available, so you do it again from the 40 to 50 day mark. So the immune response to HIV initially contains the HIV replication, but it soon slows down. So how does it contain? It has about 1 to 5% of the body's cytotoxic responses directed against the HIV epitope. It produces high levels of antibody. However, the total antibody IgG produced, 5% specific for the HIV envelope. Most antibodies do not neutralize the envelope for the HIV because it is so well selected, and there's just so much variety due to reverse transcriptase. There is high levels of envelope glycosylation. 50% of GP120 are carbohydrates, so this makes it again difficult. And there is some virus-bound carbohydrates, GP120 oligomers, actually do not stimulate a strong enough of an immune response compared to free circulating monomers of GP120, but you don't see that many circulating free monomers of GP120. So unfortunately, the antibody response isn't very effective. However, initially it does establish somewhat of a virological set point. What do I mean by this? So I mean that it reaches this asymptomatic state, also known as the quasi-state, in which the rate of viral production equals viral clearance. And we consider this virological set point, and we measure this. We know when this is reached when the RT, reverse transcriptase PCR viral RNA from the virus in the blood has reached an undetectable load of less than 50 copies per mil. Okay, so now we're going to talk about pathogenesis, and specifically what does HIV do to avoid the host immune response. So first of all, it has lots of sequence variation thanks to the reverse transcriptase enzyme, which makes a mistake for every 10,000 nucleotides copied. And the immune selection of the most favorable binding of proteins or gag proteins. The highly replicative rate of 1 billion virions per day also means you just get lots of different binding capacities. And again, the variation fosters escape from the cytotoxic lymphocytes. And this variation is the greatest in the envelope variable regions. So those are the variable loops outside the GP120 glycoprotein. And additionally, there is altered antigen presentation. This is because there's a downregulation of MHC1 by VIF, VIPU, and NEF, which are again the accessory proteins that do not play a vital role in HIV replication, but do play an important role in HIV pathogenesis. There is also the transcriptional activation by VPR, which plays a role in cell growth and nuclear import of cDNA. Additionally, there is the loss of effector cells, particularly CD4 T cells and macrophages. There is a loss of CD4 T help thanks to VIPU and NEF, which downregulate its activity. But again, this loss of effector cells is due to many reasons. Number one, there is the direct destruction of infected cells. Number two, there is the indirect destruction of uninfected cells, because infected cells may express the CD4 surface molecule, and then this fuses with uninfected cells, forming a synthesia or fused cells, and this triggers an immune activation, which again lyses the cell. Another reason why there is clonal exhaustion of CD4 T cells is because the tight junctions are lost in the gut mucosal barrier and other areas, which means microbial products can leak into the circulation, and the presence of lipopolysaccharides stimulates immune cells through pattern recognition receptors, which then activate and eliminate cytokines, which then stimulate inflammation, tell CD4 T cells to enter cell cycles, and then again get infected and die, tell CD8 T cells to become trapped in the lymph nodes, and B cells make autoantibodies. So all of these responses, all of this chronic immune activation, it causes immune cell depletion, dysfunction, aberrant lymphocyte turnover, and sometimes even organ system dysfunction. So clearly not ideal. And over here we can see some of its effects on not just the CD4 T cells in terms of loss of effector cells, but the neutrophils, B cells, macrophages, and NK cells. So macrophages reduce their phagocytosis or chemotaxis and killing because they receive less T cell help, and T cells, they produce less help, but they also have decreased cytotoxic lymphocyte for other viruses, thus explaining the vulnerability of HIV patients to opportunistic infections. B cells, they do produce autoantibodies, but they experience again a reduced capacity to kill bacteria, just like neutrophils. NK cells also downregulate their function, which means our ability to deal with cancers also downregulates. This is why in many HIV patients you see cases of Kaposi sarcoma. Now, another HIV-1 strategy is to live in immune privileged sites such as the brain and testes, which are free from attack by the immune system, and you can also reach a latency stage that is not detected by the immune system by resting in resident memory T cells, macrophages, and astrocytes. Okay, so these are all of the things we're going to be we have learned today. I hope you found this video useful. If you would like more information, or if you would like to share some of your knowledge on HIV, please feel free to comment. Thank you. Bye.